on behalf of our institution, CIIT Institute of Technology, we are pleased to have Dr. Deepak Chauhan, FRQS, TransMedTech Excellence Fellow, University of Montreal, Canada. He completed his bachelor's degree at the University of Allahabad in 2009 and master's at IIT Bombay in 2012 and doctorate degree at IIT Bombay in 2018. He acted as a visiting professor fellow in the University of Texas, Rio Grande Valley, Texas. Assistant professor at the National Project Implementation Unit, Madhya Pradesh. He prioritized himself with the continuous innovations in the field of biomaterials and cancer biology and edited by 10 patents and 5 trademarks. He likes to work as a team and manage research projects, which has led to more than 50 peer reviews, high impact for other papers involving rapture and ACS. And he has interest in technology translation from bench to bedside with experience and working with different CROs like Biochemical research organizations, which helps in starting a phase 1 clinical trial. With this short introduction, I am handing over the session to our guest, Dr. Deepak Chauhan. Thank you, sir.
Good morning, students, and I thank all the administrators to welcome me. So today I will talk about the nanotechnology. So the topic of my talk is nanotechnology changing the life. So I will start with the introduction of the nanotechnology and like how it is affecting the life of the people. I'm having some trouble. Just a moment. Okay, so my talk consists of this outline that I will talk about the nanotechnology in therapeutics and nanotechnology in diagnostics. So nanotechnology in therapeutics, mainly it is used for the treatment of the cancer and other disease. In the cancer, nanotechnology is used like magnetic hypothermia, photothermal therapy, photodynamic therapy. I will talk in detail in my coming slides. And the recent applications of nanotechnology is like a wound healing and the controlled release of the drug. Like wound healing takes longer time, but if we mix the drug with some nanoparticles, we can heal the wound very quickly. And like in a conventional mode, we take tablets at particular time, but if we make a nano formulation of it, the controlled release of the drug can be done, and we don't have to take the medicines at a regular interval of time. And in a diagnostic, as we go for the computer tomography, and there is not enough contrast, but if we do the CT with the help of some nano contrast, and then we can see the veins at very low X-ray exposure, and we can also increase the contrast in MRI imaging and other imaging modalities. So and also in the diagnostic, nanotechnology is being used for the detection of the biomarker, like the early diagnosis of the cancer, and like in a COVID also, it is being used for the fast detection of the SARS-CoV-2 virus, and also helpful in making the gas sensors, for detecting the diabetes with just the breath of the patient. But I will go in detail about the nanotechnology in the treatment and diagnosis of the cancer. So, as we all aware of that, there is a continuous rise of the cancer population, both in the male as well as in female. And in India, the conventional treatment of the cancer include the radiotherapy, surgery, and chemotherapy. And we all aware of that the side effect of these conventional therapy, like in radiotherapy, hair loss, diarrhea, mucositis, sterility, fatigue, anorexia, and surgery like the body pain, body image pain, and the higher chance of the relapse. So many problems. And chemotherapy, all we aware that how much patients suffer after the chemotherapy, like it's the systemic toxicities and nausea, vomiting, hair loss. It's a long-term problem the patient face after the chemotherapy. Uh, but in India, the problem is not only the side effect of the cancer treatment. The problem also is the poverty. Like in India, more than 2 million cancer cases are detected every year, and out of them, only 10% are able to afford the treatment, because the current treatment of the cancer cost around 4,000 to 5,000 USD. So most of the patient drop in between. And although there are some charity based hospital, like these are the photo of the of one of the very famous hospital in Mumbai. I will not tell the name, but this is the condition of the cancer patient. They are lying on the footpath outside the hospital. They don't have money to get a room, and this is the condition of the cancer patient in OPD. So like they are waiting for the treatment, and they are not getting the treatments. So in this charity hospital, so by the time patient number comes, it is spread to the other body parts, and patient usually die with the painful death. So, to address these problems, the new treatments are coming up and researched out, like magnetic hypothermia, photodynamic therapy, and photothermal therapy. So, in magnetic hypothermia, what happens? The magnetic nanoparticle are injected into the tumor, and with the help of the magnetic field, the heat is generated, and that heat kill the cancer cells. In a photodynamic therapy, the reactive oxygen species is generated, and that kill the cancer cells. In photothermal therapy, it's a light absorbing nanoparticle is injected into the tumor, and then the NIR light, that is the near infrared light, is irradiated on the tumor, and the heat is generated. That heat kill the cancer cell. I will show you one video, and that will explain what is the photothermal therapy and how it is revolutionizing the treatment of the cancer.
Oral laser therapy is a device for the precise particle-guided thermal destruction of solid tumors. Our near-infrared absorbing particles, or aeroshell particles, are infused into the bloodstream. The particles are designed to circulate in the blood long enough to accumulate in the tumor. The rapid growth of most tumors will result in a poorly formed blood supply, resulting in holes large enough for particles to exit the bloodstream and accumulate in the tumor, but not the surrounding tissue. Particles that do not accumulate in the tumor are eventually removed from the blood by the liver and spleen. After the particle infusion, the final step of the therapy is applied. A laser fiber is inserted through the skin near or into the tumor, and laser energy is applied in one or more illuminations. Particles absorb the laser energy and convert it to heat, resulting in the temperature rising to a level sufficient to destroy the tumor. The tumor will subsequently shrink.
So this is the photothermal therapy, which is currently in a clinical trial in the US and other developed countries. Still, it hasn't come to the market, but people have started working on it to make it more better. So I also started working on photothermal therapy and to make it more better and side effect free. So actually, I started working on this during my PhD, and so what I did, I also developed a photothermal agent and named it tocophotoxyl. So this photothermal agent was prepared using a polymeric nanoparticle, and the polymer I choose is a PLGA. So PLGA is a polymer made up of the lactic acid and a glycolic acid. So when this polymer degrades, it degrades into the lactic acid and a glycolic acid, which get absorbed in the body. So it's a biodegradable polymer. And on this polymer, I attached very small gold nanoparticle to make it light responsive. And the gold nanoparticle, what I attached on the surface of the polymer is 2 to 3 nanometers. That is very small. That easily gets renal cleared. So this is the biodegradable photothermal agent I developed. And after developing it, I did many biocompatibility studies. Then I injected into the mice that was bearing the tumor. So I developed a tumor model, mice tumor model, and injected this photothermal agent. And
Sorry to
then,
interrupt, Deepak sir.
yes,
Sir,
ma'am.
your slides are not moving. That's the thing.
Hello, ma'am.
Yeah, your slides are not moving, sir.
No, it's moving.
Noise is there.
Yeah,
Oh,
now it's moving, sir. Previously it was not moving.
now it's fine.
Yeah, fine, sir. Thank you, sir.
Okay. Um, okay. So after preparing of this photothermal agent, I injected into the mice tumor model. That is the mice having the tumor. And this photothermal agent very efficiently accumulated at the tumor. And after that, I irradiated, I shine the laser light, and the heat generated, that heat ablated the tumor, and so efficiently that the tumor completely gone. Like we were not able to recognize whether it had the tumor or not. And we also did the CT imaging. CT imaging, as you all know, that it's a computed tomography. So with the help of this photothermal agent, like we were able to do the CT imaging very with a very high resolution. Okay, that all I will tell. So this is the synthesis. Actually, it's more like a technical part, maybe like more boring, but I will try to explain it. So this is how like the nano, like you cannot see in a light microscope. You need a electron microscope to see it. So because it's in a nano range, nano like the virus, like virus range like 2 to 3 nanometers. So this is also like around 100 nanometers. So you cannot see in a light microscope. So after the synthesis of this photothermal agent, I saw it in an electron microscope and check its whether it's a biodegradable or not. So I kept this nanoparticle like for a month, and after a month I again did the TEM and found that it degrades very beautifully. Like after a month it degrades. So like for the nano, like people are concerned about that it goes to the body and doesn't come out. But this is not the case with this particle. It's a biodegradable. Okay, so after the synthesis, I put it for the several biocompatibility studies, because if you are synthesizing something for the healthcare, the biocompatibility is very important. So I did like a hemocompatibility study where I incubated these nanoparticles with the human blood, and check whether these damages the RBC, red blood cells or not. So it was found that these are completely biocompatible. It didn't have any effect on the RBC. And I also did the MTT assay. MTT assay is a kind of assay like to determine whether the enzymes inside the cells are active or not. So it showed a good biocompatibility even in MTTSA, and the reactive oxygen species is also very important, because the size of the nanoparticle is in comparison with the DNA and protein. So like there is high chance they may generate the reactive oxygen species. So it was found that they don't generate any reactive oxygen species. So after finding it very biocompatible in the in vitro mode, I moved to the in vivo biocompatibility. So in the in vivo biocompatibility, what I did, I injected these nanoparticle into the mice and took out the major organs after different time period, like 1 day, 4 days, and 28 days, and did the histopathology. Histopathology is the cross sectioning of the tissue and compare it with the control. So here the control is like the mice which was not injected with anything. So it was found that all the organs showed the normal signatures, like they they didn't have any adverse side effect due to the particles. And I also did the serum biochemical analysis. It's a kind of a blood test doctor used to prescribe. So what I did after 28 days of the particle injection, I took out the blood from the mice and did the serum biochemistry. So here the enzymes, like the markers for the kidney function, liver function, all were found to be in the normal range, like in a control. Okay, and hematology, everything was fine. So these particles found to be the very biocompatible even in vivo studies. So after determining the in vivo and in vitro and in vivo biochemical uh, biocompatibility of the particle, I move to the photothermal properties. So photothermal is like the property of the nanoparticle to generate the heat with the help of the light. So what I did, I tuned the absorbance of my nanoparticle in the NIR region. It start getting absorbing the light in the NIR region, not in a visible, in the NIR, and that is 750 nanometers. So and that I tuned by increasing the thickness of the gold shell around the polymeric nanoparticle. So after tuning the peak, and then the laser was shine. So when the laser was shine, the temperature raised like 60 degrees Celsius within 1 minute of the laser irradiation. So that is very efficacious photothermal agent that we experienced. And so like within 1 minute it was going to the temperature up to the 60 degrees Celsius. So what I did, then I incubated, added these nanoparticles with a different cancer cell line, like the breast cancer, cervical cancer, oral cancer, different type of the cancer cells, and laser was irradiated. So within like 4 minutes of the laser irradiation, all these cancer cells died, and the you see all these cancer cells showed almost same percentage of the death, because it is a physical therapy, not a biological or a chemical one, which may be affected by the biology of the cell, because it's a physical therapy. So it doesn't care about the resistance of the cell to the drug or any other treatment modality. So after getting the promising result in the in vitro photothermal experiment, I moved to the in vivo photothermal therapy. So what I did here, I developed the tumor model, mice tumor model. So mice tumor model I developed, like I injected the cancer cell in mice and develop a tumor in the mice. It's all I did after taking the ethical clearance in the institute. You cannot do like this. Okay. So after developing the tumor, then I injected my nanoparticle intratumorally. So and then laser was irradiated after injecting the particle intratumorally, and then laser was shine, mono wavelength 750 nanometer laser was shine, and within like one shot of the laser irradiation, the tumor completely gone. It's gone, and even after a month, it didn't come back. This mice completely remission of the cancer. It's cancer free. So this was the intratumoral injection, giving the injection directly into the tumor. But there are many cases where you cannot give the intratumoral injection, and then you have to give only the intravenous injection. So in this case, you need to prepare the targeted nanoparticle. So what I did, I developed a targeted nanoparticle by attaching a folic acid, because cancer cells express more folate receptors. So if you attach the folic acid onto your nanoparticle, they will preferentially uptaken by the cancer cell. So these nanoparticles were preferentially uptaken by the cancer cell, and after here also, and after it got accumulated there, then again a laser is radiated, shine the laser, and within 2 shot of the laser is radiation, the tumor completely ablated. It's gone. Like the mice completely remission free with free with the cancer. So and it didn't come back even after the 6 month follow up, and this mice lived as comparable to the control. And yeah, and we did other studies to confirm whether these nanoparticle accumulated in the tumor or not. Like we took out the tumor and did the sectioning of the tumor and see whether the gold nanoparticles are there. It's all other control studies we did. Yeah, and the other thing is this particle, as I told, also work as a wonderful contrast agent. Like we can increase the contrast during the CT imaging, computer tomography. So I injected these nanoparticle into the tumor and did the CT. So as you can see in the video, you can see the depth of the tumor. So that is very important, because the solid tumor, the surface tumor we see, but we don't know how deep is this. So we can see the depth of the tumor in the 3D CT, and that can help the doctors to optimize the dose of the laser irradiation, dose of the particle, concentration required, and everything. So so we have developed the technology, and then we looking forward to start the clinical trial. And but before that, this particle need to be synthesized in a GMP lab. So GMP lab is a type of the lab where the medicines synthesized, because we cannot inject the nanoparticles synthesized in the lab to the humans. They need to be synthesized in a clean room in a GMP lab. And in India, now the nanotechnology GMP labs is coming up, which is taking forward. So hopefully we will able to synthesize our formulation in a GMP lab, then we'll able to start the clinical trials. Currently it is handled by the Neomics Research Organization, one of the CRO. And yeah, thank you everyone. Hello.
Yes, sir.
Hello.
Yeah, am I audible, sir?
Yes, ma'am, you are audible.
Yeah, one student is asking a doubt. Can we use this nanotechnology in our society? He's asking.
Yeah, nanotechnology, in our society is being used. If you go in a Mumbai airport, I have my experience. There in the washroom, it's a water free toilets. They have built up, because they have added the silver nanoparticle. They have made a coating of the silver nanoparticle to the toilets, and so they don't need water, because silver nanoparticle can kill the bacteria. And so they don't need water in the urination. Mm. Yeah, and there are many applications of nanotechnology. People are synthesizing nanoparticle with the help of the medicinal plants, and to attach the molecules, the active, the beneficial molecule to the nanoparticle, and reach the target, reach the beneficiary, the organs where it is supposed to be there. Because nanoparticle usually work as a carrier. It can take your drug where you want it. If you take the medicine, it will go everywhere in the body. Like if you take the medicine for the headache, but it will also go to the liver, spleen, kidney. It will not only go to your brain, but if it is attached with like nanoparticle or some targeted agent, you can make them go only to the place where you want. So like you will reduce the amount of the drug needed. So ultimately you are reducing the toxicity. Like you don't need to take that much amount of the medicines. And I see that many like nanotechnology is coming up every, like in automotive, like in lubricants, it is being used and to increase more to decrease the friction of the lubricant to make it last longer. And it is being used like for making the technologies better and advanced. Currently now it's coming up is now, like from the lab it's coming to the market from the society. It's coming slowly, but it's coming up.
Fine, sir. The next question is, can we include artificial intelligence into these nanoparticles?
Sorry ma'am, what's your question?
Can we include artificial intelligence into these nanoparticles?
Yeah, like in this COVID, people are using AI and to see like how this nanoparticle is affecting the virus and everything. Like they are actually using the AI in the imaging of the patient, and after injecting the nanoparticle and this AI, I think it's more useful. And actually I don't have much idea about this AI, but this nanotechnology, like they are making a multifunctional, like they are using it like they are adding a transmitter and everything, like nanotransmitter, and it gives the signal from inside the body, and that they are using like many functionalities you can do with this nanoparticle. So this is like one of the application I told in the healthcare, like how it's how useful in the healthcare, like in the treatment of the cancer. But there are many applications people are doing in computer science and electrical engineering, mechanical engineering, every, I think all domain of the engineering.
And if anything has to be predicted, definitely there will be an analytical part. So they are asking what kind of programming language can be used for analyzing this nano inputs.
What kind of programming language can be used? That is
Yeah.
the question.
Yes. Yes. Yes.
Actually, I see that this Python people use mostly in this like biomodeling, something like in biology simulation. They learn the Python mostly, I see.
Okay, sir. Is there any additional courses in nanotechnology for the freshers? Is there any additional courses?
Nanotechnology additional course, like I see that now many IITs and other colleges like are running the online courses on nanotechnology. Like recently I see the swam, the portal swam, like there are also many courses on nanotechnology. And that's there. Actually, biotechnology, I see many students are of here biotechnology and biomedical engineering. So now the, like everything is merging up. Like earlier, like the research was in like the basic discipline, but now everything is merging up. Like biotechnology is merging up with the nanotechnology, and biomedical engineering is coming up with the other domain, like the computer science and electrical engineering. So everything is like merging up. So the students who are here biotech or electrical engineer, like they can see their future like in a interdisciplinary science. Because I remember that in our lab, like there were the guy from the electrical engineer, computer science, and even mechanical engineer were also there. So it's all about merging.
Sir, we have biomedical and biotechnology and agri also. So one of our agriculture engineering student is asking, can we use this nanotechnology in agriculture or agriculture engineering field?
Yeah,
Can you
yeah.
provide
In
details?
in agriculture, the people are also using nanotechnology, like for adding the fertilizers to the field. Because if you add just a fertilizer, it leaches away. But if you add with the nano formulation, so it stays longer in the field. It will not leach away fast, and you can also target the weeds with the help of this nanotechnology. There are many applications in agriculture. Actually, agriculture is like a very nowadays is very uh, hot topic, because people didn't know that they can apply this advanced technology to agriculture also, but now they are applying it. So it's a very hot topic.
Fine, sir. One of our biotechnology students is asking, can we use this nano in stem cell?
Sorry, I missed. Nano stem cells.
Uh, can we use nano in stem cell?
Nano in stem cell. Yeah, actually, I told you, like everything is emerging up. Like stem cell is a part of like biotechnology, and nano is nanotechnology. But now people are using this nanotechnology like for engineering the stem cell, like for delivering the gene inside the stem cells, or for for managing its protein expression by interfering with the translation mechanism. So, like this nano technology helps in a uh, better delivery of moieties into the stem cells. So many things. Now even like found that our body also generates the nanoparticle, that is the exosomes. So that is the endogenous nanoparticle. It's found that uh, like uh, uh, the cells communicate each other with the help of the exosomes, and these exosomes are like 40 to 100 nanometers nanoparticle that is made up of the liquid, very small endogenous nanoparticle, and that carry the message from one cell to other, and they communicate. So
And one more question, sir. While comparing to the other technologies, nanotechnology is higher in cost. Is there any chances for reducing the cost in future?
sorry, ma'am, your voice is breaking. Can you please repeat?
Yeah, while comparing to the other technologies,
Mm-hmm.
nanotechnology is higher costly, right, sir? So, is there any chances for reducing the cost in future?
Yeah, I got the question. Nanotechnology is not higher in cost. It's just because we don't have the enough resources, or like we are not so advanced in this. Like we are just beginning. Like initially, if something comes, it's like in a higher price, like our mobile data in India. Earlier it was very costly, but now it's very cheap in the world, even cheapest in the world. So I think when the several like market players will come into this field, it's going to be the very cheap.
So, that's it from student side, sir, questions. So thank you, Dr. Deepak Chauhan, sir, for the interesting session and the informative presentation. You have said about the nanotechnology in the diagnosis and many things you have said, and you have given clarification for our students' questions, and you have
Yeah,
given the solutions for that also. Thank you so
yes,
much for spending
anytime
your
anytime they can contact me on WhatsApp. WhatsApp or LinkedIn, like they can ping me like if they need any guidance in biotechnology, biomedical, or nanotechnology. Because I see that biotechnology students feel that they don't have anything to do after their BTech, and but it's not like that. There are many things you can do. Like I did my master's in biotechnology, and then I entered in biomedical engineering, nanotechnology, and I did many things. Like, and even my friends did many things after the BTech in biotechnology. So it's not like that. Just you like if you don't know, then you maybe you don't have the enough information. Like you are not in a good environment. You're not in a with the good friends who are aware of this field, because it's a very rising field and you can do many things, and currently everything is connecting up. Like it's not like that you did in biotechnology you are nowhere. It's not like that, because the company, like the vaccine, drug manufacturing, they need interdisciplinary people. It's not like
Fine. Thank you. Thank you for spending your valuable time with us, sir.
that. Thank you.
Yeah. In this occasion, a special thanks to Dr. K. Mahalakshmi Ma'am, Dean KITC IIII, for organizing this webinar. I wish to express my gratitude to our management, principal, vice principal, deans, and HODs of various departments, faculty, friends, and students.
Mhm.
Thank you, sir.
Thank you very much for inviting me.
Yeah.
Thank you.
The students,